front of you at the moment. So what I'm going to guide you in over the next few minutes is taking this, which as you can see, a lot of the work's done for you. The writing's done. You're actually going to end up only jotting a few things in your journal. It's very user-friendly. We just need to piece this together. This is actually going to become a foldable. So if you would, take out your scissors, and we are simply going to cut around this first page. So cut around the circle. So what does this circle represent? Who can identify this? Very good. This is the cell cycle. You will cut out both circles. So once you finish that first one, you can move on to your second one. Miss Neighbors, could you go around with a trash can and collect their scraps? So what you should have in front of you once you've done your cutting looks like what you see on my screen. We should have both circles cut and present. I want you, once you have your cutting done, to locate this particular circle that you see on my screen. Locate this particular circle that you see on my screen once you are done cutting. I do want to remind you that G1, S, and G2 are all a part of what we collectively call interphase. After interphase, a cell moves into mitosis. Once mitosis is complete, a cell undergoes cytokinesis, where it truly cuts and breaks apart into two identical daughter cells. Who can tell me what part of the cell is actually separating during mitosis? It's one part, one organelle is actually duplicating itself. Who knows what that organelle is? Starts with an N. It's the nucleus. Very good. So go ahead and let's get this piece. You're going to glue it on your journal just like you see mine on the screen on your next blank page. Glue it down. Yeah, a lot of them are. Once you have it glued down, locate your second circle and your scissors. It is important on this step that you follow my directions explicitly. You need to not only listen, but also watch so that you do not make a silly mistake, okay? These lines, we're about to cut them all the way up to the circle at the center, but we will not cut through the circle at the center. We are not cutting these off. We are simply making them into flaps. So I have not cut all the way through. I can now flip up each one of these sections of this second circle. Once I have this step complete, you'll notice that the circle I glued in my journal has a little area that says glue here. Put a little bit of glue in that area at the center that says glue here. I'll be putting glue right here. Pretending to glue. Only putting glue here in this center. It's a whole lot of glue. All right. Now, this next step, I have to line it up perfectly. If I don't line it up perfectly, my foldable will be wrong. So we are going to take our second circle that we cut, and you're going to hold it just like mine is held. G1 will be at the top left, S will be on the right hand side, G2 will be at the bottom. You can check to see if you're putting it right, because where you cut should line up with a line on the circle behind it. So you're going to match yours up. And then you're going to press to make sure that that glue adheres through the paper. Here is one that is complete. So now when I lift up S phase, I see DNA replication. DNA is copied. When I lift up G2, I see organelles replicated. It's preparing for mitosis. If I fail this check, what do I get? Cancer. Then in mitosis, I see PMAT. Cytokinesis, I see cytokinesis. Basically, the cytoplasm is splitting. And in G1, cell growth, normal metabolism. If I fail this checkpoint, what do I get? Cancer. So what you have in your journal should look like what you see right now on my screen. We're going to add a tiny little bit of notes, and then we're going to take a brain break. So we're going to open, actually give me a fist of 5. Who is caught up to this? Not the writing, just getting everything put in your journal. Are we done, or do we need a minute? Good, everybody's ready. So we're going to flip up our S flap. Under the S flap, it says what steps. I want you to put, we unzip with helicase, unzip with helicase, and polymerase builds. Unzip with helicase and polymerase builds. Both of those end in the suffix ase. So what are they? What was our warm-up over today? Starts with an E. In what?
Enzymes.
Thank you. So all of these here are enzymes. Polymerase. Polymerase. Polymerase.
Again, anytime you see a word end in ace, that indicates it's an enzyme.
Up here, I've given us our apples and trees and cars and garage to remember. A bonds with T, G bonds with C. I've put that my 5' to 3' strand is leading. That's the one that polymerase can copy continuously. It's not going to struggle.
Can you push it down?
Yes, I'm sorry. And then I put that my 3' to 5' strand is lagging. Do y'all remember the crazy name we call the chunks that get made on that strand? It was someone's name. He was not American, and it started with an O.
No.
Those are called Okazaki fragments. That's what I said. Okazaki. They were in our Cornell notes last week. Was that last week? Yeah, that was last week.
On Friday?
Maybe, yeah, it was Friday. All right. Getting down the remainder of the information under S phase. G1, S, and G2 are all a part of what? Look at your foldable. It's labeled. G1, S, and G2. Yes, they are all a part of interphase. The cell spends most of its life in interphase. I don't need to write anything on G1 and G2. That information's already been given to you. Open up your mitosis flap.
Can you scoot it up a little bit?
We are now opening that mitosis flap. Here it has our acronym PMAT written. Notice I've written the words for those. So by my P, I wrote prophase. By my M, I wrote metaphase. By my A, I wrote anaphase. By my T, I wrote telophase. So if you remember our hand signals, it is prophase, metaphase, anaphase, telophase. And that's exactly what you see here in this drawing. DNA is visible, but still central in the nucleus. DNA is lined up at the middle of the nucleus. DNA has broken into two separate portions. It's being pulled to each end, and DNA has now made two complete nuclei. So this is prophase, metaphase, anaphase, and telophase. All of that is happening in the nucleus. It is not the whole cell, it is the nucleus of the cell. And then the last thing I want you to get is that cytokinesis is actually just when the cytoplasm splits. And like Miss Neighbor showed you yesterday, that's when you chop. We now end up with two identical daughter cells.
Is that a C or a O?
Huh?
Twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork.
There's that first enzyme.
The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
And that's where we'll stop. If you will, open your journals up to the next blank page. Through our exit ticket yesterday, there is conception about the structure of DNA. So we're going to take one page of DNA structure notes today. This is why I had you grab a couple of different colored pens. I will be changing my ink color as we go through these notes. It's going to help you stay organized mentally if you're able to do the same. We are calling these DNA structure notes. DNA structure is on the heading of our paper. Be sure we are following along and on task. So the first thing that I'm going to do is draw my 5' to 3' backbone for my DNA molecule. So it's going to start, my backbone is going to include my phosphate. There's a phosphate bond. It's going to attach to that sugar. That sugar is going to come out. We're going to have another phosphate bond. And then another sugar. I'm going to label this here. Phosphate bond. This strand is 5' to 3'. So if this were that DNA molecule undergoing replication, this is the side that would be able to replicate continuously. The polymerase wouldn't have to make chunks. It would be able to go in order. So, attendance. It's Miss Stoley. Yes, ma'am. Miss Neighbors, could you bring your device up here? Because if I log in here, we'll lose all of the notes. So this right here, I'm going to take a separate color. I'm going to draw a box around it. And I'm going to label this is the backbone. So I'm going to say phosphate, bond, and sugar, backbone of DNA. But wait, there's more. In addition to that backbone, I've got to have my genetic information. So I'm going to draw this now. These are my bases. Go to the La Vega website. It's, go to La Vega Staff Toolbox and go to Skyward. And then Rachel.store. And then I'll put in my password. What? Lies. If you don't mind, just... Yeah. All right. That is one side of my DNA. So I'm going to change my ink color. I got to look at the other side. Because remember, it's a twisted ladder. So I have to have two sides that are connected by rungs. So I'm going to draw my phosphate bond. Notice something here. My sugar is upside down now. Y'all see that? Yes. So it's an upside down house. I got another phosphate bond, and I've got my other upside down sugar. So it's flipped. It's going the opposite direction of the other one. That's because this side is my 3' to my 5' side of DNA. Again, these are my phosphate bonds. Off of these, I will have my bases. But now, I have two sides to this ladder that are my backbone, right? But is that ladder joined? Is it joined down the center yet between my bases? They're still separate. So I'm missing something there. I've got to come in here with another bond. So this here is actually a hydrogen bond. And that hydrogen bond can be broken by an enzyme. Who remembers what enzyme can unzip DNA up its hydrogen bonds? I heard it, I think. We just saw it in the video. Yes, sir.
Good job.
Come here. Yeah, you. Good job. Helicase can unzip that DNA up the hydrogen bonds. Another thing that I want you to take note of
As long as you know that they're side by side.
is the fact that these bases in the center, those are my genetic information. Those are my apples in trees and cars in garage. So my outside edges are my backbone. My middle bases are my genetic information. They are held together by hydrogen bonds that can be unzipped by helicase. Adenine with thymine, guanine with cytosine. Give me a fist of 5 on having this down. Most people are writing, that's fine. I've got one more drawing, actually 2 more small drawings to make. So I've got to clear my screen before we can continue. So I'm going to wait for everyone to get caught up before I continue. All right, is anyone still writing? Make this quick so we can be done. All right, I am going to now draw that is DNA, where half of it is still coiled and half of it has opened up. We are going to put a couple of enzymes in here.
Whatever that is.
Oh, that is so ugly. What am I doing?
All right. I want you to know, just basically, that, and we haven't gotten deep into this process yet, but DNA can make something called mRNA. We'll talk more about that later. And that mRNA can make something called, you should recognize that word, a protein, which is one of our biomolecules. In this diagram that I've just drawn, I'm going to label the parts here for you. So this is a DNA strand. Half is still coiled up, half is unzipped and open. It was unzipped by the enzyme helicase. It unzipped DNA up those hydrogen bonds between the bases. I have 2 enzymes here that are building the DNA. Do y'all remember what it's called? Polymerase and helicase. So polymerase is building that DNA strand. If this one is 5' to 3', this is my leading strand, so it can go continuously. If this one is 3' to 5', this is my lagging strand. This is where I get my Okazaki fragments. We're going to go ahead and wrap up with this drawing today. I do want you to know that DNA replication occurs in S phase of interphase. What phase does nuclear division occur in? When does my nucleus divide? What phase of the cell cycle? What's the letter that represents it? M phase, and that stands for the word mitosis. When you finish this drawing, you may go ahead and close up your journals, put up your pen, glue, scissors, and journal boxes, and then Miss Neighbors is going to lead you in our exit ticket for today.